DNA is a long thread-like molecule that provides a blueprint of instructions for your body's 50 trillion cells. Your DNA is kept in structures called chromosomes, which are located in the nucleus of cells. If you took all of your DNA from every cell and stretched it out, it would be long enough to reach the sun and back about 4 times, covering more than 744 million miles. A gene is a region of DNA that contains a specific code. The code is written in a 4-letter alphabet of chemical bases or nucleotides. A gene sequence tells the cell how to make a specific protein molecule. Proteins have distinct shapes and functions, which allow them to control many aspects of cellular anatomy, physiology, and metabolism. Proteins include enzymes, receptors, peptide hormones, and structural building blocks. The complete set of genes you carry is called your genome. Humans share the same set of an estimated 20,000 genes. Variations in those genes explain why no two people are the same. A variation in a gene sequence may alter the structure, function, or amount of the protein that is made. Genetic variations account for many traits, including hair color, blood type, body weight, and nutrient assimilation. In medical genetics, mutations are defined as rare variations that affect less than 1% of the population, and they typically produce a predictable health outcome. In contrast, polymorphisms are more common and have milder and sometimes beneficial effects. Polymorphisms are often influenced by multiple environmental factors. The most common type of polymorphism is a single nucleotide polymorphism, or SNP. A SNP is a simple typo in which one nucleotide is replaced with another. Your genome contains an estimated 10 million SNPs, but only a small number of them have any effect on the protein that is made, and therefore are not medically relevant. The terms SNP and mutation are commonly used interchangeably, but it is important to remember that SNPs are common, modifiable, and unable to independently cause health conditions. Genetic testing enables medical professionals to identify unique opportunities and challenges. Since many SNPs influence nutritional needs and responses to common dietary and lifestyle factors, genetic testing provides important clues that aid in personalizing diet and lifestyle protocols. As the field of nutritional genomics is still evolving, it is important to focus on SNPs that are well-researched, clinically relevant, and responsive to interventions. To interpret genetic test information, it is important to understand common nomenclature, which is shown in this example of the MTHFR gene. This gene has a common SNP at position 677 of the gene sequence, where a C is replaced with a T. The C is considered the normal allele, and the T is considered the variant allele. The term allele simply refers to the different versions of the same gene, the one containing C or the one containing T. SNPs also have identification numbers called RS numbers, which are useful when searching the online medical literature. Genotype refers to how many copies of the gene, if any, contain the variation. Everyone has 2 copies of most genes, one from each parent. If neither copy has the variant, the genotype is called wild type, which is often considered normal, although there are exceptions. If only one copy contains the variant, the genotype is called heterozygous. If both copies are affected, the genotype is called homozygous. Dietary, environmental, genetic, and epigenetic factors can influence the effect of a SNP on a person's physiology. For example, the C677T SNP in the MTHFR gene reduces the stability of the enzyme encoded by the gene. This enzyme converts dietary folate to its active form. By maintaining adequate folate status, carriers can mitigate the functional effect of this SNP. Pure Genomics makes it easy for healthcare practitioners to identify and address common SNPs that are well-researched, clinically relevant, and nutritionally actionable. Learn more at puregenomics.com.